In the first place, I want to say that it's a pleasure for me to present our work. I want to thank the organizers for it. And what I'm looking at is a registration node for large serial section microscopy datasets. And the project is embedded into a biological project together with Albert Cardona from ETH Zurich and Volker Hartenstein. And this project is extracting particular microcircuits of interest in the Drosophila Instar CNS, which you can see here in an optical microscopy image where the nuclei are stained with VAPI. And just to tell you a bit more about that structure, this is the full nervous system. It has 300 times 200 times 150 microns. It's well ordered, so we have here the ventral nerve cord, which actually is the of the larva, and here's here are the lobes, and what you see here as these different intensity is exactly the cortex, which consists of all the neurons that then project into what is called the neuropile, where all the is on. One can see it a bit nicer in this beautiful schema from E2 and others. And here the brain lobes, these are the thoracic segments. Now, in order to identify really the microcircuit exclusively for microscopy, because the find is more than 100 the optical. At the time, the video has abdominal segment about 80 microns that provides you both the field of view that is to identify these structures. Is transmission of Cardona together with Vetter from that. One of these abdominal in 409 transversals with a single image size as applies. It looks actually sort of the ventral channel, distal connections, and then rates of projecting are okay. Now, electron microscope is built of bunches that focus the electron beam, a grid that holds the specimen, so the series section. Finally, this ejected phosphor screen, and the sections themselves are stacked with heavy metal atoms, and these heavy metal atoms scare the electrons, so the heavy metal atoms are enriched. You see black where they are not. So this scene from the top, it is a grid. It's a copper grid. Here is a plastic sheet, and on a ribbon of serial sections that you cut under the size of, like in our application, you can serial sections into one grid. So you have many of these grids and now you image them, and the image looks like the brain, not the segment, but that wouldn't matter. Cameras in microscopes are either just the conventional that get by default. They have a resolution. For it, it's not enough to gain the real images. I'm sure for each section as the series data to be. This is what you're telling the micro is something like that. The distances are very small, so it cannot find these locations. More from one section to the from a relational code that handle a situation like this. And here you can see that this is also a manual process and also inserting one grid is completely known. Significant distortion during these so no, let's first at the images. This is registered data. We have clean sections that are nicely during the operation. There also occur some excess, and you have some area setting down of the staining solutions. Is heavily distorted, introducing even this in a more formal transformation from the into location in the final images that we see. In this model, we still consider that each section parameter, and we assume that the thickness of the section is well. So we actually look just at a 2D problem in that case. First place with gamma term, unknown deformation that was applied during sectioning. Then we have a beta, so this relies on a section. We have an unknown deformation, which is a heat-induced transformation during imaging. So you make the image, heat up the sample, and it's deforming. So we have some deformation in there. Then we have a rigid component introduced by inserting one grid after the next. We have a rigid component one section to the next, and of course this sensational component from one to the next. And as last deforming factor, a lens distortion in the electron microscope is completely removable on the hardware level. To handle this, of course, we decide to simplify this a bit. We join rigid component to one rigid component, and also try to approximate this transformation that applies to the section by allowing a rigid individual transformation individual for each single image tile. So all single images can rotate and not just translate and shift and stretch a bit and so on. For this, we have now the problem that is a template-free regression, so we don't have any reference to project that into. So it's a differential application, and for this we need corresponding image content. And in order to handle the artifacts that I showed you before, we need something which is very robust within the sections. And this is sort of okay because we show the same image content actually. But across sections, if we assume that the signal changing function is identity, which is actually not true, but compared to the thickness of a section, it's very close to changing not at all. We need something that can just ignore these little artifacts. And for that, we found that that was developed computer vision scaling development actually performs very well. With this, we have to figure it a bit first to explain you how first I find points of interest well in two dimensions. And it identifies the possible scales in the image from the original pixel. And these blob structures identify gradient histogram and the scale and the rotation span frame to extract a local descriptor from local invariant to scaling, rotation, and translation. Acting just the intensity patch in this image, the images that are at different places, the version is completely rotated, looks already very nice to our eyes. But to compensate for further artifacts, what they do in the even smaller local groups, this patch into insensitive to alignments and stuff like that, right? So this works very nicely within the section. Cross section is not valuable, but mainly because this very small application in computer vision of and the images more or less everything looks just like that. The first step is to just view, make the descriptor larger. We don't need real time. It's a bit more time consuming to manage them, but make the descriptor 512 descriptor better results. These are corresponding features. Regression, the form, we see a section, you see small differences here, this kind of changes, it's here and like that. Here we have two sections that are related by including something else or deformed a bit. Now, here and there, depending on the parameters the skeleton. Just using the local descriptor. This case at matches, and you can already buy that. I'm just leave wrong. Actually, situation where we have 90 movement sample consensus tests many models from and checks for all other regarding this randomly selected model. It's smaller than a given threshold later, and if it's below the threshold, there's inliers, and then counted as outliers. The two sections, the corresponding pairs, it chooses random 2 points. For which transformation you don't need more, and tests all the others. And here right side, you use error distribution, and for all the bad matches, this error distribution is and it's bars, and for find and distribution, it's both also how you the model. The winner is. So still we see, we choose here, there was a big tool, slightly too information that we accept. And we do any inlier and align just that. But and we can see this decision contains, if you see from the shifted center of the area. We now fix the iterative or marginal tail of this error distribution. So you calculate the model, estimate the errors, and reach all point correspondents that have a transfer error larger than 3 times of the error distribution. Means it wouldn't remove too many good matches. Like that, we see here free mean. We have your matches, and if you this is a nice example. Very that. Match has a very simple, but it's the wrong one, so it should be here, right? But because it looks very nice, but it's close enough for RANSAC to accept it as a good match. So all the regression model is getting better and better. And finally, we end up in this most of the largest parallel, which is a good hint in this transformation is correct. Okay, now finally, we made of assumptions. So we approximate information with transformations. We assume that the signal changing function across T, which is not, and we have errors from correction because they may be slightly mislocalized. And so what we have to do in order to get a reasonable solution is to minimize the global error. This is it's just very simple. So we minimize the overall displacement or corresponding pairs, all tiles that share point pairs with respect to feature transformation. And for now, we let this intuition be identity. This we do in an iterative scheme that each iterative step, we just estimate with the closed form solution the best transformation of each individual tile, then update the whole system, keep the rest fixed to the next time, and so on, and as we do until the overall error converges. And if you do that for multiple sections, it's actually like that. This was just a single section. You can add more and more and more, but you have always to for the whole global error, so you end up with a minimum, and all sections try to fight against being deformed. Okay, so the remaining part is the distortion. Is done by Verena Keinig. One can do these steps iterate, nothing changes anymore. Of course, you have to focus just on the illustration, because if you apply this distortion to correct for a cross section, wrong. And eventually it's all a closed form solution, so you keep the rigid transformation, apply this polynomial transformation of marks within these times, and that gives you an evolution for it. The image usually does something like that, and if you do that on a montage, you see here a full montage, and here zoom in the thing but mostly the okay. Optimization and the rigid transformation had been necessarily introduced to just render the images by just putting these rigid effects like this. So because this configuration is just optimal for the critical sections, register then estimate a smooth file as control point to determine to this configuration. And the configuration, this is a smooth transformation field corresponding to it, and it's using transformation as described in this. If you look into significant fixed. So an approximation, it's the data set, but it's a whole thing, and it will segment all these single more, and here you can the last, which actually requires very end. This you can open themselves. But here we see a synapse, and here a synapse, but you also see that this is the size of dendrite. And it, okay. Like that, and not original illustration that we knew until we try to do it in this way, is just to you take 1 second, the first, the third to the second, and so on. Very continuity between two is totally fine, but in our application we also want to raise this and less. And in the top of this slide, you see this presentation, and here just comparison with the bias line that just forward propagates the transformation. And what is here is forward propagating transformation is so and then in order to fit these sections together, that structure was there, and in the end you result in a which is not desirable if you really want. This is the result of our approach, showing we see that the section direction of this complete would be like that, but it's a bit tilted off to align your sample in a way that you exactly meet a. Okay, last but not least, the is an application that everyone can use it into the Trachy M2 framework, which is designed for large serial section mosaics and annotate them with the control displayed in a tree with many annotations to 3D objects and 2D objects and things like that. And all this the image data distribution Fiji, and it's fully open source to that project. And Fiji is located in Pacific. Also, it's still institute in Dresden. More and all these things that can be reused on all these people because they contribute to the development in track and completely different. That is a record identifies you from MIT in the distribution doing correction application, and Stephan Saalfeld of folk stacks matching local. I want to thank people as society and our institute. Then my boss, RANSAC, and all the lectures, so currently people. Albert Kuhn is in he and does whatever he wants. Volker Harten for biology a lot, and is highlight transformation models and and event. This code was developed in the main this guy here. Thanks for your attention, and I hope we can go for a few questions. Yeah.
If a particular action is folded, do you interpolate that particular folding area after stitching, or you leave it as it is?
So discontinuities in this application are not explicitly handled. It's a continuous transformation. In case of a section fold, it would stretch the region around the section fold. Yes. You could make. I mean, when the section fold is actually going from one edge to the image, there can be two images, and then you have the discontinuity explicitly displayed. And in track, this is what you're leading. So if you have an image where a fold goes from right to left, you just split that image, cast one image, one image, the other, and have the discontinuity expressed. But it's not expressing discontinuities explicitly. That's deep.
Can you talk briefly about sort of the 3D space, more to do with the distortion of 2D, and then if it's a conformal local stack when you encounter distortion or tension, how you're doing it for 3D stitching?
In 3D stitching, let's back. Yeah. So, in application, it's not handled. This is just solving for switching this line. Right? So, in this application, we are with a function that there's no distortion. But in small, sorry for this line jumping, I have no idea why. We found that using just an affine transform match all of about 1 pixel. So that's hard to have to correct for after one microscope they're using, right?